Today we're going to be talking about protein structure. Proteins are a little bit different to carbohydrates, so it's important that you've got a good grasp of what's going on with their structure. In the course, you'll find that the structure is split over more than one lesson. So what we're going to do here is we're going to tie everything together so you get a bit more of a cohesive understanding of what protein structure is all about. So proteins are built of amino acids, and our amino acids all have the same general shape. Okay, so there we have nitrogen, of course, because we're talking proteins. So we have our NCC backbone. Then we have a double bond O, single bond OH. We have an H. We have two more H's. And we have an R. So what are all of these bits and pieces? COOH, this here is our carboxylic acid group. And on the other side, NH2 should sound a little bit familiar. This is the amine group, where the amino acid got its name, amine carboxylic acid. See, it all starts to combine together, and the more linkages and things like this that you can find, the more it is so much easier to remember things. Also, when you're understanding the levels of protein structure that we're going to get into just now, we need to know what's an amine group, what's a carboxylic group, where are they in this amino acid that we're talking about. So last one, H is an hydrogen, straightforward and simple. He's not going to do anything weird. The weird one is this R. Now the R stands in for a variable group. Okay, so it could be as small as an H, or it could be big and complicated with a whole ring structure going on. Okay, we don't need to know the details of all those different groups, but we do need to know that R is where our variable group sits, and that variable group, we have 20 different variations, which gives us then 20 different amino acids. So all of the proteins, everywhere, all the ones we study, all the ones we don't study, they are all made from only 20 different amino acids. And it's all about how those different amino acids are joined together, the order of them, the sequence of them, the next level of bonding that happens, that gives us that enormous variation in protein structure. Because we have only 20 different amino acids, but we're building them into proteins. So a few things to be aware of, okay? No, we don't need to know all 20 R groups, we don't need to know all 20 different amino acids. Okay, you're gonna bump into some familiar ones over and over again. And there's 3 of them that you want to keep in mind from the beginning. Okay, glycine is an interesting one. Glycine is your smallest amino acid because its R group is just H. So instead of having an R here, we would just have an H. And there is our smallest, neatest, tidiest little amino acid. And that, of course, has knock-on effects of the structure of any protein that has glycine in it, particularly if there's lots of glycine. Okay, so glycine has the smallest R group, which is just the hydrogen. Then the other 2 come as a pair. They are cysteine and methionine. And these 2 are funky because these are the 2 that have sulfur in their R group. One of the bond types that we're going to talk about just now is a disulfide bond. Now, of course, the only time you could have a disulfide bond is if you have sulfur in your R groups. So that's why cysteine and methionine are, again, important ones to keep in mind. Okay, so basic of a protein structure is amino acids, the building block. We should actually have probably written that somewhere, isn't it? Because this is actually amino acid structure, the building blocks of those proteins. So what happens next is that we've got to bond these amino acids together to build a polypeptide, and then we're going to do all sorts of funky things to that polypeptide to turn it into a protein. So if we think about that bonding between amino acids, we would say, okay, well, we've got some familiar looking OOOs and H's, and maybe we're heading in that same vein of condensation to build and hydrolysis to break, and we absolutely are. So you can take two amino acids and you can stick them together and you make a dipeptide, then you make a tripeptide, then you make lots of them for a polypeptide. So that's the approach that we took in carbohydrates. We're going to take it slightly differently in proteins, because it's not just about linking them together into a polypeptide. It's about what then happens further from there. So amino acid is our basic building block. Then we have primary, secondary, tertiary, and quaternary structure. Okay, so primary structure is all about that linking into a polypeptide. It's the peptide bonds. Between amino acids. Between does not have 3 E's. Okay, secondary structure. Here is the start of our changing things. Okay, we no longer just have a polypeptide, we're going to add in some hydrogen bonds. Okay, but these hydrogen bonds are all about the COOH and the NH2. So you can see now why that carboxylic acid group and amine group are so important, because that's where the understanding of the secondary structure comes in. Tertiary structure is all about bonds between R groups. So you see now we've used the basic condensation, we've used the amine and the carboxylic, and then in our tertiary structure we're talking about this variable group. So it's really important that you've got the aspects, each 3 different pieces of that amino acid, so you can start building up. When it comes to quaternary structure, it's all about associations. Prosthetic groups, multiple polypeptide chains, prosthetic groups and multiple polypeptide chains, okay? And it's all held together again between bonds between the R groups. So it's got a similarity to tertiary, but quaternary is a whole different beast of its own, okay? It's also an optional level. Many proteins don't have quaternary structure. Many of them just go to tertiary structure and then that's it. Okay, so we're going to now take a little bit of time to look at each of these levels of structure in a little bit more detail. So we've got a thorough understanding of what happens to this collection of amino acids as we move through the levels of structure. So starting at the beginning. Primary structure. So primary structure is very simply the formation of our peptide bonds. Okay, so we have formation of peptide bonds. It's a condensation reaction. Okay, we're just joining them one to one to one to one to one all the way along. Okay, but what's important is 2 things. Important features: which amino acids are incorporated. So the particular amino acid and the sequence of amino acids. Because if you think about it, each amino acid has a different R group. Now that R group ultimately is going to be used in tertiary and maybe quaternary structure. So if we have different amino acids, we then have different options for different layers of structure. So both the particular amino acid, the type of amino acid that's used, and then the sequence. Which sequence do we go in? Do we go 2 of this, 1 of that, 3 of that, 1 of that, or do we go 4 of that, 1 of that, 5 of that? Okay, and all of that is then going to have a knock-on effect along the structure. So we can think of primary structure, okay, as forms a polypeptide. A long chain of amino acids. And it can be illustrated, they would be stuck together because they'd be bonded, okay? But that's it, just a string of amino acids. It's not actually a functional protein yet. Because after primary structure, we've now got to have some fun. Okay, first level of fun is secondary structure. And here we have folding and coiling of our polypeptide. So we've taken our string and now we're doing things to it. Okay, we've got 2 different types of secondary structure that we've got to consider. First one is an alpha helix. Okay, so this one is a corkscrew shape. So it would look something like this if you're looking for it in an image. Okay, and this is hydrogen bonding between the CO of an amino acid and the NH of another amino acid 4 places ahead. So, hydrogen bonding between CO and NH 4 places ahead. So we would have our primary structure, right, of our amino acids. And what we're saying is this amino acid here is going to form a hydrogen bond with 1, 2, 3, 4, this one here. And then we're going to get 1, 2, 3, 4. So obviously if we're going to form a bond, then these amino acids are going to have to come closer together. And that's what then leads us to form this coil, this alpha helix. Our beta pleated sheet is our other secondary structure. And that is a little bit more challenging to draw. It looks a bit like this. So you've got some examples in your book, you've got some examples in your notes that are a better 3D representation of beta pleated sheet. But again, it's all focused on hydrogen bonding. Okay, here we have hydrogen bonding that's holding parallel sections together. So we have hydrogen bonds between these parallel sections of the polypeptide. So the last thing to note about secondary structure, to highlight, to hammer home, would be the fact that this is all about hydrogen bonding between amine group and carboxylic acid group. The R groups are not involved in the secondary structure. Okay, so I've had a question come through of whether the hydrogen bonds are every 4 amino acids or amino acids 4 places apart. That is a very good question. We've done every 4 here, but 4 places apart could actually be taken as, if we colored in this one, it would be 1, 2, 3, 4 blank ones, and then we'd color in that one. So that one I'm going to have to look up, and I will get back to you on. Any other questions relating to primary and secondary structure? So now we'll go on to tertiary structure. So tertiary structure, we've got to take this protein that has primary and secondary structure already, and we're going to work with it further to make it into a 3D shape. Okay, so here we would want to say that we have further folding and coiling of the polypeptide. It's only really after tertiary structure that we would refer to it as a protein. Okay, this is forming a precise 3D shape. Okay, so primary structure was just about getting those links. Secondary structure, a little bit of coiling, a little bit of folding. Okay, tertiary structure, we're now further folding and coiling to form a precise 3D shape. This is all about bonds between your R groups. So it's going to depend on what atoms you've got there within the R group, and then what kind of bonds they can form with each other. When we're talking about types of bonds, there are 4 different bond types that we need to think about. First one would be hydrophobic interactions. So if we have R groups that are hydrophobic, they'll want to cluster together. We can have hydrogen bonds, they're everywhere. Okay, remember that's a little bit of a charge difference, then we want to associate. We can even get, in these ones, a full-on charge difference. We can get ionic bonds between different R groups. So we could have a plus in one R group and a minus in the other R group, and there, of course, the plus and the minus would want to bond with each other. Okay, remember, none of these are covalent bonds. The only covalent bond that we're going to get between those R groups is the disulfide bonds. But those, of course, are only between R groups which have sulfur in them. You can't get a disulfide bond if you don't have sulfur. So we have interactions, hydrophobic interactions, we have hydrogen bonds, we have ionic bonds, we have disulfide bonds. And what we've done is we've now taken from our primary structure, we've then coiled it a bit, coiled it a bit, folded it a bit. And now we've really squiggled and wiggled and wobbled it up, and we formed a precise 3D shape. So for example, your enzymes, okay, will often have a globular shape. So there'll be a little blob, and there you'll have things like hydrophilic R groups on the outside, hydrophobic R groups on the inside, because if our enzymes are going to be in the cytoplasm, then they have to have a hydrophilic outside, or we'd have a real problem. Okay, so tertiary structure takes us to our precise 3D shape, and that is often the end for proteins. So all of our proteins have that primary, secondary, tertiary. Some proteins go on to have that quaternary structure. And we said at the beginning, quaternary structure is all about associations. Okay, so associations between what? We could have multiple polypeptides. So if we have a polypeptide like this, and then we have a polypeptide like that, and these two together make one protein, it has quaternary structure. We could have a polypeptide with a prosthetic group. So something that is not based on amino acids, like a heme group in our hemoglobin. So there would be our polypeptide with a prosthetic group. This now has quaternary structure. It's not just one polypeptide that's been folded. Our last option combines all of the above. We could have multiple polypeptides with prosthetic groups. Maybe one, maybe more than one. Okay, that's why hemoglobin is so wonderful, because hemoglobin, you've got 4 polypeptides and each of them has a prosthetic group. So you've got like all possible levels of quaternary structure being indicated. So for example, there's our protein, 2 polypeptides with a prosthetic group. That would then classify as quaternary structure. A lovely thing about quaternary structure, same bonding as in tertiary. So it's all about those R group interactions that are giving us the quaternary structure. So it's beg your pardon. It's the hydrophobic interactions, the hydrogen bonds, the ionic bonds, and the disulfide bridges. And that is what holding our polypeptides together, or our polypeptides and our prosthetic groups together in some way, that it then makes a single unit, a single functional unit. And that's the thing to remember as well, you know, when it comes to a protein with quaternary structure, if that protein doesn't have its quaternary structure, then it's not a fully functional protein. You wouldn't take one polypeptide out of hemoglobin and say, oh no, that's a functional molecule, because it's not. Hemoglobin has its 4 polypeptides and its 4 heme groups, and that is what makes a functional protein. So a protein with quaternary structure has to have its quaternary structure to be a functional protein. But it all goes back to and depends on that primary structure, those peptide bonds linking those specific amino acids. Because if we don't have those specific amino acids in the specific sequence, then we don't have the R groups in the right places to then make the tertiary quaternary. Okay, so we need the sequence of amino acids making the primary. We need then that starting of the alpha helix and the beta beta sheets in the secondary, and then we take that further with the R groups in the tertiary, and then potentially working with associations for the quaternary. So it's 4 levels of protein structure, 3 that you've got always, 1 that you've got sometimes, but all 4 levels are critical in forming a functional protein, depending on what that protein is.